Hello, everyone, and welcome to today's panel presentation titled Revolutionize Your Lab Work with Connected Electronic Pipettes with Live Q&A. This webinar is a part of the 7th annual event in the Drug Discovery and Development Virtual Event Series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Sartorius. For more information about Sartorius, go to www.sartorius.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You will also notice that the speaker has provided a survey located to the right of your screen. This survey will be available during both the live session and the on-demand period. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar from the lobby within the event. Now with that, let's get started. I'd like to now present today's speakers, Sandra Söderholm, Application Development Scientist, Liquid Handling at Sartorius, and Joni Ake, Product Manager, Liquid Handling at Sartorius. For a complete biography on our speakers, please click on the Presenter tab from the menu on the left of your screen. Sandra and Joni, you may now begin your presentation.
Hi, my name is Joni Ake from Sartorius.
And hi, my name is Sandra Söderholm. I'm also from Sartorius. And today we're going to talk about connected pipettes and how they can help you in the lab. So please go on, Joni.
Okay, and today we are going to look generally the advantages of electronic pipette and the advantages of connected electronic pipettes. And then we're going to delve into the more application-specific benefits you'll get. Pipetting is one of the most common and important tasks performed in labs. It's very general utilized. They can often make or break your experiments, and pipetting has been around for a while. Around 1950s, we switched from pipetting by mouth to micropipettes after they were invented. We moved towards multi-channel pipettes, especially in the microbubble plates in 1970s. And in the late 80s, Biohead, part of Sartorius today, introduced a electronic pipette with the DC motor that really helped and sped up the working. So electronic pipettes have been going strong around 30 years already, and they are getting more and more popular all the time. And today, as I said, we're going to look how they will help you and your work. Laboratories face a lot of different challenges. For instance, increase or pressure to increase the efficiency while maintaining reliability and repeatability of results. Pressure to reduce the time to results. And at the same time, we definitely need to uphold the well-being of employees. Well, while considering your laboratory's productivity, consider the tools used. They can make great impact. If you have a lot of sample throughput, if you have a low sample throughput, a good mechanical pipette might be everything you need. An electronic pipette will help increase your throughput while remaining the flexibility of a mechanical pipette. You can semi-automate some protocols, and it's a good in-between step when you can't justify robotic systems. Electronic pipettes definitely give you the same flexibility as with mechanical pipettes, but they increase the throughput and help you to semi-automate some tasks. Even if a lab has most of the liquid handling needs met by a robotic system, there is very commonly still certain things you need to do with the manual pipettes. All the exceptions are typically handled by manual pipettes. Certain sample handling still performed with manual pipettes, cherry picking, etc. So there's a place for various tools within a lab. A lot of laboratories we've seen, they have dozens of pipettes, but typically only a few tend to be electronic ones. There are exceptions, of course. We've seen labs that are fully equipped with electronic pipettes because of the benefits you get, but very typically only a few of them. We've seen, especially when preparing the 96-384 well plates, users tend to move towards electronic pipettes because they reduce the human error and increase the throughput. Another reason you might move towards electronic pipettes if in case the employee has suffered from some sort of injury due to heavy pipetting with mechanical pipettes. But there are some other reasons, well, that you might consider switching to electronic pipettes, and this is what we're going to discuss next. Electronic pipettes, generally, they make lab work more ergonomic. They reduce the user-to-user variance. They create more repeatable and more accurate results. They can be faster and safer. In addition, if you talk about connected electronic pipettes, they enable pre-programmed workflows and user instructions while performing sample preparation or assay preparations. And this is what we're going to discuss next with a few examples. Electronic pipettes, they are very ergonomic to use. And there are a couple of points to consider here when choosing a handheld pipette. First is the height, weight and height of the instrument. Shorter pipettes, they reduce the need to lift the arm higher, especially if you work in a laminar flow cabinets or safety cabinets. There's a limited amount of space for you to work in. So the shorter the pipette it is, the better in terms of ergonomic it is. Then the weight. Let's take Picus 2 for an example. It's a lightweight electronic pipette. It's as light as a mechanical pipette, so it's easy to lift around and use. Even during long periods of time, it doesn't strain the user. Next is the operating forces. Here in the slide, you see an example of accumulative forces required to operate pipette through the day, per cycle. Because as it's fully electronic, everything from aspiration, dispensing to tip ejection is electronic. So the user only needs to click a button, as compared to mechanical pipette, where the tip ejection is the heaviest part that strains the user. You'll see the huge difference that the user or the force is required to perform a typical cycle. And as you know, typically you don't perform one cycle per day, but you might pipette many hours a day. So the difference in the end of the day is quite significant. Then user-to-user variance. That's one of the big advantages of an electronic pipette. As the piston movement or aspiration and dispensing is done electronically, the user thumb movement doesn't impact the results. So because pipettes, or electronic pipettes, they reduce the assay-to-assay variance per person, but especially they reduce the operator-to-operator variance. Here is results from a study performed by Sartorius, where we divided a large group of people into three categories. There were unexperienced pipetters, moderately experienced pipetters, and then experts. the data shows the different groups when it comes to mechanical pipettes. It's easy to see the experience level of a person, especially if you adjust the pipette to lower volumes. So, for example, with 1000 microliter pipette, when you're pipetting 500 or close to 100 microliter, the difference become greater. But when the same users did exactly the same experiment with the electronic pipette, then we tend to, or the results show us that the difference between the user groups gets smaller. So this study showed us that the electronic pipettes definitely help to reduce the variance between the users and brings all the users to expert level. Then if you compare mechanical and electronic pipettes, the electronic pipettes do have capabilities in form of different pipetting modes. They definitely can help you to speed up work and reduce the physical stress required to get the work done. For example, multi-dispensing and sequential dispensing modes in electronic pipettes, they significantly speed up the work when you are filling a plate, for example. Here, multi-dispensing is perhaps the most used mode with an electronic pipette, and can be definitely one of the biggest time savers. It can be typically 3 times faster to fill in a microwave plate, or if you compare it to a mechanical pipette. How it works is you aspirate a lot of the liquid into the tips, and then you dispense smaller aliquots, typically like 8 or 12 times, depending if you have 8 or 12 channel pipette. If you want to fill in a 96 well plate, you might aspirate 800 microliters and then dispense 10 times 100. Saves a lot of time and reduces the need to move back and forth. Especially, it can be very helpful if you combine it with a plate tracking functions. So if you're working with the microwell plates, they are transparent, and typically you handle a small amount of liquids, and they tend to be also transparent or very lightly colored. So it might be a challenge to remember in which well you need to pipette next. Or if you've ever been in a situation where a colleague walks by and asks you something or tells you a funny story, then you totally can get lost where you need to pivot next. But a plate tracking function can help you to remember, okay, I need to be in C8 next, for example. And the electronic pipettes, they do make the lab work safer with these advanced features like calibration reminders, locking functions, and the connected electronic pipettes, they can provide you instructions, pre-programmed protocols. Here on the screen, we have a 96 well plate example where you are working with a single channel pipette, and it tells you where to aspirate, in this instance from a tube, and then you need to dispense to a B1 well. It gives you very visual guidance what to do. But even alone, the electronic pipettes with the reminders, you never forget the calibration date, and you can lock the pipettes until they've been calibrated, or you can also use the locking function in case you accidentally contaminate the pipette or it's been dropped. You can lock it and prevent anyone from using it until it's been serviced. Then speed settings. This is again one of the major differentiator between electronic and mechanical pipette. Electronic pipettes, once you set the speed is constant from the morning to afternoon and also to next week and the next week and so on and so on. Especially if you are working with foaming liquids, viscous liquids, surrogate media. something like that, it really helps you to control the speeds very repeatably. You can completely avoid the foaming by using, for example, a multi-dispensing or reverse pipetting and very slow speed. You can avoid foaming completely. And it's from all the pipetting you are going to do with the electronic pipette. So you don't need to be that careful and introduce a lot of stress towards the user to control the manual pipettes and the pistons. But electronic pipettes, you click button and it uses exactly the set speed very constantly and repeatedly. Final slide from me, updating the pipettes. The connected pipettes, the manufacturer can provide updates and also upgrades. So you can always be sure that your purchase a pipette today and you can enjoy the updated features down the line and maybe new features even if the manufacturers bring something new. So the investment you did once, you don't lose the value, but you get everything, all the new benefits that the manufacturer is bringing out. And next, Sandra will start to talk about the benefits of an electronic pipette in the application. She'll also discuss about the new possibilities that these connected pipettes bring to work and the workflows.
Thank you, Joni. So I will start here with talking about the pipetting then in life science applications. So even though pipettes are used every day in labs, pipetting practices is not a topic that laboratory professionals tend to care about. So many, they are self-taught and have never received a formal pipetting training during their career. So as a result, many scientists may not know when to use forward or reverse pipetting or how the pipetting technique can affect reproducibility of results. So next I'll show some examples and discuss why electronic pipettes are beneficial in life science workflows. So a bit about reverse pipetting, especially for challenging liquids. So please apply reverse pipetting technique when pipetting challenging liquids. For example, foaming, as Joni already mentioned, viscous or volatile liquids, or if you're also struggling to pipette small volumes. So the picture here to the left guides you through the reverse pipetting process with a mechanical pipette. First, you press the piston button all the way down to the second stop, as far as it goes. Then you go to the vessel with liquid, aspirate it by releasing the button completely. Then you go to your target vessel, dispense by pressing to the first stop, so not all the way down. After this, there is some liquid left in the tip, which you can discard by pressing all the way down to the second stop. So reverse pipetting, it's also possible with electronic pipettes. The steps that I just explained, they're just automatically done for you. So in reverse pipetting, an excess liquid is aspirated into the pipette tip, as I mentioned, but only the volume that you set on the pipette is dispensed. So liquid is then lost due to adhesion to the plastic tip. It's compensated from this excess liquid amount. Reverse pipetting also prevents aerosols and bubbles to form, which is important in case you work with cell cultures. One important thing to notice when using reverse pipetting is that your pipette tip have to fit the total volume plus this excess volume. This is extremely important to keep in mind when you are using filter tips, because you do not want the liquid to touch the filter inside the tip, as this leads to reduced accuracy, loss of sample, and also possible contamination. So here you see a graph of an experiment where HeLa cells were pipetted with an electronic pipette to 96 well plates using either forward pipetting mode with blowout, which generates a bit of bubbles. It's seen here as a blue line. Or then using reverse pipetting, which creates no bubbles here in magenta. So the results, they propose that bubbles, they interfere with even distribution of cells and absorbance-based measurement and generate larger deviation between replicates. Bubbles tend to also have a negative impact on cell health and can interfere with microscope-based measurements, for instance, live cell imaging. So keep in mind that multi-dispensing mode on an electronic pipette, it works similarly to reverse pipetting, and it's also recommended for cell seeding. So you can combine pipetting steps into a complete protocol or workflow on most electronic pipettes. Here's an example of a protocol where mixing steps were combined with pipetting the target volume when seeding cells, here in graph, the first graph, plate 1, or then no mixing steps were applied before seeding cells, so in graph B, plate 2. Each dotted line is cell confluency in one well over time. So for plate 2, where no mixing was performed or programmed between the seeding, the variation in cell confluency between wells was higher than for plate 1, where you had a mixing programmed in the workflow. qPCR, it was a topic on everyone's lips during the COVID-19 pandemic. Electronic pipettes can save more than 50% of time it takes you to pipette a qPCR assay on 384 well plates compared to mechanical pipettes. The time saved is mainly thanks to the multi-dispensing mode, for example, when pipetting master mix to the plate. The plate tracker mentioned earlier by Joni guides you so you won't pipette into the wrong well. In case of sample replicates, you save also time by multi-dispensing the RNA or DNA samples. So pipetting practices, they can greatly affect the reproducibility of sample preparation and ultimately the outcome of, for instance, cell-based assays. So in cell-based assays, as an example here, removing the cell culture media from microvell plates with too high speed can cause cells to detach, for instance. Adding details regarding how pipetting steps should be performed, they are often omitted from published protocols, even though these can be very crucial for how reproducible the assays are in the end. So in this example, the modes, the speeds, and the advanced functions on electronic pipettes were added to a lentivirus titer assay protocol to ensure high reproducibility of the assay. The readout of the assay was performed with the live cell analysis system. So connected electronic pipette, they open up a whole new dimension of traceability and user guidance for sample prep workflows and pipetting protocols. With the newest electronic pipettes on the market, you're able to make workflows, for instance, in a mobile application, which then automatically selects the most optimal pipettes for the workflow that you have available, sends the pipetting settings, the volume, speed, and mode to the connected pipette via Bluetooth. It also automatically does the switch between the different pipettes depending on the step in the workflow. Additionally, gives the graphical overview of each step in the workflow. So all these features are particularly helpful when working with long and very tedious workflows. On the slide, there's an example of a serial dilution workflow. It is currently instructing the user to dispense 200 microliter to well C1 on a 96 well plate. Once the user presses the operating button on the connected electronic pipette, the task will happen, so the liquid will be dispensed, And then the pipette will automatically set to the volume speed mode required for the next step, or the app will just automatically connect to another pipette if this is required. Yeah, so yeah, thank you for your attention.
Yeah, please ask any questions. We'll do our best to answer them.
Thank you so much, Sandra and Joni, for your informative presentation. We will now start the live Q&A portion of the webinar. So to our audience, if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the right of your screen. Now let's get started. Our first question here asks, what else besides choosing an electronic pipette can I do to address user-to-user variability?
Thanks, Kaylee. User training is one of the key aspects of reducing user-to-user variability. That's really the cornerstone of everything. Pipetting is still a very manual work, even if you use an electronic pipette. So having all the users trained into the topic is very important. Not to forget regular maintenance and calibration of the instruments.
Great, thank you, Joni. I have another question for you here. Can you use one speed on your electronic pipette for all your workflows?
I wouldn't advise to do so. The speed setting can greatly impact the results you get, especially if you're talking about liquids that differ greatly from water properties. Like highly viscous liquids, I would slow down the speed as much as needed for it to completely get into the tip and get out from the tip. Also, this is very good advice for the foamy liquids. For some liquids, a faster speed might yield good results, and obviously it's going to be faster when pipetting as well. So choose the speed according to the liquid.
Great, thank you. Now, you mentioned filter tips, but what else can one do to prevent contamination?
Using reverse pipetting technique, in this way, less aerosols are created because there is no blowout at the end of dispensing. Changing tips after each pipetting step, and absolutely every time you touch something else than the liquid or the vessel you are supposed to touch. Regularly cleaning the pipettes, and before and after you use them, autoclaving the pipettes, or at least the lower ends, if possible, and then storing the pipettes in a stand. This way they are out of the homes where there's no dust accumulation, less liquid accumulation inside the tip, the tip cone of the pipette.
Great, thank you, Joni. Now it looks like we have time for one more question here, so we'll wrap up after this one. This final question asks, can we always use reverse pipetting?
Technically, I think you could use it. It might be a little bit slower to perform, so that's why a lot of the users don't use it for all the pipetting steps. So choosing the most suitable pipetting technique based on the liquid, the situation where you're in, and application is still advisable. So forward pipetting is a very good technique. You don't always need to use reverse pipetting or multi-dispensing. You can utilize the standard pipetting, but for those difficult to pipette liquids, definitely a reverse pipetting is advisable.
Great. Well, that will conclude our session for today. So I want to thank you, Joni. Do you have any final comments for our audience?
Thank you, everybody, for tuning in. It's always a pleasure to talk to the audience, and hopefully you learn something new as well.
Yes, absolutely. Well, I want to thank you again, Sandra and Joni, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, Sartorius, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until February 21st, 2025. Labrets will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.